so my name is Hamid Najafabadi. I'm from McGill University. So I'll dive right into it. I don't need to tell this crowd that genes are expressed in the form of different RNA isoforms. There is alternative splicing. And also, I don't need to tell you that it's really important to quantify isoforms. I know that a lot of times we just quantify the level of expression of genes, but then if you want to study anything related to RNA processing, alternative polyadenylation, RNA splicing, RNA export, or even degradation, we really need to measure the abundances of RNA isoforms. And how do we do this? Well, we have short-read RNA sequencing, fairly available right now, cheap, and as a result, we can get really high coverage. But on the other hand, the reads are short, and because of that, they're ambiguous. They can map to multiple different isoforms. So they're not always very informative about which isoform they come from, and therefore it's difficult to measure the isoform. We have long-read RNA sequencing, which solves the ambiguity problem, but then on the other hand, it has other issues. It's still expensive, more expensive than short-read RNA sequencing, and even as the prices are going down, both short-read sequencing and long-read RNA sequencing, they're going down together. So there will always be a cost advantage to short-read RNA sequencing, and because of that, we're going to get lower coverage for the same amount of resources. And as I will talk in some of the slides, there are also other issues with long-read RNA sequencing, existing technologies, including length biases, GC biases, and other artifacts such as retruncation. So there are advantages and disadvantages to each of these approaches. And really, I think the optimal strategy is to try to combine these two approaches, combine their strength in order to get optimal isoform abundance quantification. And our solution for integrating these two types of data is this tool, MPACT, multi-platform quantification and analysis of transcripts. Developed by Michael, a graduate student in my lab. This is the preprint, if you're interested. The preprint has a lot of the things that I'm going to talk about today, but not everything. So there are also some new stuff that is not in the preprint. So what is MPACT? MPACT is essentially a generative Bayesian model. It tries to describe how the transcriptome profile of a sample gives rise to what we have observed in the data. In short-read RNA sequencing, we observe short reads. We can group them into these observation units, which are essentially the sets of reads that have the same type of information about the same types of transcripts. If you're familiar with pseudo-alignment, for example, equivalence classes are a type of observation units. So these are reads that can map to multiple different isoforms in the same way. And what MPACT does is to describe how the transcriptome profile can lead to a specific observation unit profile. And a key component of this framework is this matrix P, which is the matrix of conditional probabilities of the different observation units given a specific transcript. So given one unit of a particular isoform, how many reads do we expect from that isoform to be assigned to a specific equivalence class? The framework is generalizable. So it's not just for short-read RNA sequencing, obviously, because we're talking about multi-platform quantification. What changes from one platform to another platform is the shape and definition of these observation units. So for long-read RNA sequencing, for example, we can, if really the reads are unambiguous, then we can consider individual transcripts as being the observation units. And when the observation units are different, this P matrix also becomes different, this matrix of conditional probabilities. But the idea is that we can use the same framework to describe the process of generating reads from different platforms. So that gives us also a way of inferring backwards, starting from the observed data in short-read RNA sequencing or long-read RNA sequencing, and figure out what is the transcriptome profile that optimally describes the data that we have observed. So if we only have short-read RNA sequencing, we have one platform, we can use this framework to infer RNA abundance from short-read data alone. If you have multiple platforms, short-read plus long-read, the same concept applies, but now we're trying to come up with one single transcriptome profile that explains the data from both strategies. So one sample, short-read RNA sequencing, also long-read RNA sequencing. What is the underlying transcriptome profile that generated this data? So does it actually work? Let me start with just short-read RNA sequencing analysis in bulk tissue data. We've done various types of benchmarking. I probably won't get a chance to talk about all of them here. More are in the preprint. So we start with synthetic data because it's always the easiest. We know ground truth from which we are synthesizing, generating synthetic data. And we show that with short-read data alone, MPACT outperforms state-of-the-art methods that are very popular for quantification of isoform abundances. In real-life data, we've tested many different things. We have our own spike in RNA control experiments, for example, that have been sequenced with short-read RNA sequencing, data from different publications. This is one example based on sequin spike in RNA control. So these are our synthetic RNAs with known concentrations that are spiked into samples, and then we sequence the sample, and then we try to recover the known concentration of these synthetic RNAs. With Callisto, which is very popular, we get a good line, but also we get these things that look like dropouts. So these are sequin transcripts that essentially Callisto tells us have zero abundance. We use MPACT, we don't get this, so everything kind of looks straight. It's not just about absolute abundance estimation. We can also use it for differential abundance estimation, and it works really well. So this is an example where two different sequence samples with different proportions of different RNAs are being compared. We're essentially trying to come up with differential log-fold change in abundances. Again, I'm comparing Callisto and MPACT. Callisto has these weird outliers that are not really well behaving. MPACT doesn't have that. So we also do very well in differential abundance estimation. And more benchmarking comparison, like consistency between estimates from short-read data and estimates from long-read data, different kinds of spikings, different kinds of samples with a lot of variation in RNA abundance with little variation, little differences between samples. In all of these cases, we see that MPACT is the state of the art in abundance estimation. So that was about bulk RNA sequencing data. Can we also do this with short RNA sequencing? And this is actually a very challenging question. I don't think people have really been hopeful about estimating mRNA isoform abundances from short-read sequencing at the single-cell level, because most of the approaches for single-cell analysis have this very huge 3' bias or 5' bias, depending on what kind of technology and kit you're using. So a lot of reads that we get are, for example, from the 3' end of a transcript. And that's problematic because, well, first of all, we don't have any information from the middle part of the RNA where a lot of the exons and exon junctions that are informative about which isoform the read comes from are located. So we don't have information about that. But I think even more importantly, this kind of bias violates a key assumption that is made by a lot of these quantification tools, such as Callisto and Salmon and Arsen, which is that the reads are uniformly sampled across the transcript, and that really throws them off. So how does MPACT deal with this? Well, it's actually fairly simple to implement it. This is the work that Jichen, another graduate student, has done to add this capability to MPACT. And the idea is that what really changes is the P matrix. The observation units that are farther away from the 3' end of a transcript, for example, they're actually less likely to be seen simply because the technology doesn't capture them. So if we knew the weights of how distance from 3' end, for example, or 5' end relates to the probability of capture, we could inject that into this P matrix, kind of adjust the P matrix, and get something that again can be used for quantification. And this can actually be done within the expectation maximization framework of MPACT itself. So we don't need to a priori know these kinds of biases. We can ask MPACT to just estimate these biases from the data to maximize the likelihood of the data given the model parameters. So, for example, when we apply this to chromium 3' single-cell data, this is the kind of profile that we get. These are the weights that we get for observation units based on their distance from 3' UTR. It's as we expect. closer to the 3' UTR, the higher the weight, the higher the likelihood of capturing a read from that region. The farther away from 3' UTR we go, the probability goes down. So the weight profile makes sense. Do we actually do good in isoform abundance estimation? So here I'm comparing impact without considering these positional biases and impact with these positional biases against the scalpel, which is a very recent method. I think it came up a couple of weeks ago for isoform quantification from single cells. So there aren't a lot of tools for isoform quantification in single cells, which is why we're kind of limited in our benchmarking efforts. So I only have scalpel in the mix. And here we're not really comparing abundance of isoforms versus the ground truth. We're doing something more difficult. So we're looking at the relative usage of isoforms within a gene. So we're saying that of all the different isoforms that this gene transcribes, how much of the abundance comes from this specific isoform. So it's a ratio between 0 and 1, essentially. And the ground truth is bulk RNA sequencing from the same cell line that is also being assessed by a single cell. We can see that even impact without positional bias works better than this one tool that we have been able to identify, but then impact with positional biases, it really outperforms. Okay, so everything so far has been about short reads, but multi-platform quantification. So do we do better if we also include long reads? So what is the advantage of adding long reads to short read data for isoform quantification? Again, we start with synthetic data. Here I'm talking about synthetic data set with known ground truth, 30 million short reads, 200,000 long reads. Not a lot of long reads, but it's intentional. I'll tell you what's happening here. So as expected, short read plus long read just slightly does better with short read alone, because really this data set is dominated by short reads. There are very few long reads. What I want to highlight is that with even as few as 200,000 long reads, if we directly compare our short read based inference versus short plus long read inference, then we see that there are a subset of transcripts that are really affected. So their inferred abundance is really different between a short read alone and short read plus long read. These are long read sensitive transcripts, if you may. And when we look at how we do for this subset of transcripts, then we see that adding long reads to short reads really helps. So we have even negative correlation with short reads alone, but then the correlation shoots up when we add long read sequencing. And what are these transcripts? These are isoforms that come from genes with many exons and genes that are very long and genes that have many different isoforms. So these are essentially isoforms that come from genes with complex splicing, which makes sense. It's difficult to resolve the ambiguities in these very complex genes, and we need long read data. And if we do a gene set enrichment analysis, functional enrichment analysis, the top hit with a very strong p-value is neuronal systems, which again makes sense because we know that in neuronal systems, splicing plays a very critical role in shaping the transcriptome of the cell. Which is why in collaboration with Gudarzi lab at UCSF, we wanted to use impact to actually study neurons and neuronal differentiation. This is the work of a postdoc in Gudarzi lab, Benedict, who did in vitro differentiation of embryonic stem cells into cortical neurons. And then we did short read and long read RNA sequencing at different stages and used impact to infer RNA abundances. And one of the things that we did with this data is to, again, try to figure out if short read plus long read using impact works better than short read alone. So one idea that we tried, and with the help of Larissa, who was a graduate student in my lab, was to try to estimate exon usage, so cassette exons, based on our isoform-centric quantification. So the idea is to just add up the TPM of isoforms that contain an exon, divided by the sum of TPMs of all the isoforms of that gene, and that gives us the percent spliced in of that exon. And then look at differential PSI, differential exon usage, between embryonic stem cells and differentiated cortical neurons, so kind of delta PSI, but then do it with short read and long read, or with short read alone, and then see if there are any exons that are really quantified differently based on these two approaches. So these are the exons that, depending on whether we include long reads or don't, their estimated change in inclusion is really different. And then we can go in the lab and do QRT-PCR to see which one is correct. And then we can see that QRT-PCR measurement of exon inclusion really correlates strongly with what we infer from short read plus long read using impact, but very weakly with short read alone. So that was about the advantage of adding long read to short reads. But one might argue that, okay, just do long read sequencing with a lot of coverage. It will be expensive, but you can do it, and then you won't need short read RNA sequencing. So is there any advantage to short reads being added to long read sequencing data? One thing that we've noticed through analysis of long read sequencing data we've generated in the lab, PacBio data, as well as what's in the literature, is that there's quite a bit of bias in terms of transcript length and GC content that is captured by these technologies. So for example, in our hands, when we do PacBio RNA sequencing, for example, using spike and RNA with known controls, we can see that RNAs that are longer are overrepresented, and RNAs that have higher GC content are underrepresented. And if we control and correct for these biases, we actually get a much better correlation between the ground truth and what we get from the long read data. But how do we learn these biases in an experiment-specific manner? Well, the good thing is that by comparing short reads and long reads and having them together for one sample, we can again estimate the same biases very consistently. So this is why having short reads actually helps, because it allows impact to learn these biases from that exist in the long read sequencing data, again in the form of modifications to the P matrix, and make better predictions. This is another example. This one is from Oxford Nanopore, telling us that some mRNAs have almost zero abundance based on Oxford Nanopore, but when we combine short read and long read, we see that some of these are actually very highly expressed. A lot of them are long isoforms. And if we look at the individual cases, then we see that the reason is that Oxford Nanopore reads seem to be a little bit truncated. So they show profile very similar to what we get from single-cell RNA sequencing, which makes the mapping suffer to think that these reads actually come from this very short isoform and gives zero to these longer isoforms. Whereas PacBio and short read sequencing data tell us that the longer isoforms are the ones that are being expressed. And this is something that impact fixes. So I realized that I have like 2 or 1 minutes, and I haven't told you anything about machine learning, whereas the title of the talk actually starts with machine learning. So here it is. So one of the things that we were very interested in was to use these impact measurements to study the sequence determinants of isoform abundance in neuronal systems. And this is the work that is done by Ali in my lab. He's very interested in machine learning modeling of RNA splicing. One of the things that he tried was to just see whether the features within 3' UTRs of RNAs can predict RNA abundance in these neuronal tissues. We do fairly well for a subset of transcripts, very good abundance estimation. And interestingly, these machine learning predictions based on the motifs within 3' UTRs can actually predict relative isoform usage within a gene. So here, in this example, this longer transcript with longer 3' UTR is predicted to have higher abundance because of presence of these motifs that probably stabilize the RNA neurons. This one with the shorter 3' UTR is predicted to have lower abundance, which corresponds to the experimental evidence. So can this be used actually to improve the inferences of impact? So the idea is to use impact to estimate TPMs of mRNAs, train a machine learning algorithm like this, a machine learning model on this, on our sample, and feed the predictions back into the impact in the form of a prior on the abundances of the transcripts. The idea being that these algorithms, they learn these rules about what determines mRNA stability from the transcripts that have good abundance estimates, and then they can help the model to resolve ambiguities for the other transcripts for which this information is not quite available. So we apply this concept to the same single cell data that I showed you before. So MPACT estimates abundances, machine learning feeds back into MPACT new abundance estimates, and this loop continues until convergence. And then you have the same three graphs as we've seen before, but then this last one also uses this kind of machine learning prior for relative isoform usage. They're really doing well. I think considering that these are single cell data, which was not supposed to be useful for isoform abundance estimation, I think we're doing fine. So I'm going to skip the summary because it was short, and I'm sure that you remember what we were talking about. I talked about the people who were leading this project and also our collaborators. Thank you very much for your time.
Thank you. We have time for one question.
Yeah, so it was a great talk. One question I had, so you showed the advantages, right, that short read sequencing provides to the long read and then vice versa. In practice, do you find that in order to get that advantage, you need an equal amount of samples from both? Or could you have an overrepresentation of one and a bit less of the other to get to the same outcome?
Very interesting question. So we haven't done this analysis, but I think one thing that we need to do and would help answering this question is a form of subsampling or simulation where you keep your budget constant and then you try to see how you need to allocate your budget to short reads versus long reads to get optimal abundance estimations. We haven't done that, but I think that would be very informative.
Thank you very much. Okay, we're going to move on to see this